Can anyone else hear her?
No, you're on
Oh,
mute, Miss Rudisil.
Here we go. I figured out what the issue is. My bad. All right, guys, here you are. Now we're going to talk through the PowerPoint slides for this week. And then I'm going to have you guys watch the protocol video. I'll share the computer audio and it should mute me, which is what just happened. But then you'll be able to go in and watch everything, figure everything out, and kind of work through the material that way. So participation and professionalism points from now till the end of the semester are going to be based on participation in Zoom meetings. So you can ask questions in the chat by raising your hand or speaking. You can turn on your camera and nod and reply to questions in the chat and talk to your group mates in the breakout rooms, and also participate in polls if I provide those to you. So I'll also be giving participation points for things like students emailing in with questions towards the end of the semester or just in general for the week about assignments, materials, and also just letting me know in advance if they won't be able to attend lectures. So we're going to talk a little bit about gel electrophoresis. I know we hit on this last week, but this may be something that you guys aren't as familiar with because it has been a while since you've heard about it. Some of you haven't heard of it since high school. So this is a biomolecular technique for separation and analysis of macromolecules and their fragments based on their size and their charge. We did hit on that a little bit last week. So the basis of gel electrophoresis is that there is a gel-like sieve. You can kind of see this here, and this separates fragments of DNA based on their size and their charge. The pores in this gel are what help to separate the various sizes, again, of those fragments, with the smaller fragments moving further along in the gel and the larger fragments moving more slowly and staying up towards the more negatively charged end of the gel. So there are a couple of different options for the gel sieve, and it's polyacrylamide and agarose. Most people typically use agarose, and polyacrylamide is typically used for finer detail, such as when you would have tiny fragments or sections of DNA where you really haven't been able to amplify as much, and you need to really be able to see what you're working with. So again, here's a couple of steps in this process. So you start with your glass plate. You would put your combs in first, okay, and then tape with the masking tape. Then pour the heated agarose solution into the glass tape and form a solidified agarose gel. After it solidifies, you would add in your ladder, which is a basis of comparison, once it had been put in a buffer. And typically, we're going to be using something called a TBE buffer for this class. And you'd add that each well in individually afterwards, being careful not to puncture the individual components of the wells. And then you'd run the gel. And remember, we're running from a cathode to an anode. Cats, again, some people don't like them and they see them as negatives. And anodes, some people think ants are cool. So just think of it that way. So that negative end to that positive end, DNA has those phosphate groups, right? So they have that negative charge. So they're naturally more inclined to move away from that negative end towards that positive end. But larger fragments do so at a much slower pace. So pouring the gel. It comes in a powdered or tablet form, and it's heated, typically in 30-second increments. This is not something that you want to overheat. If you overwork the gel and you overheat the gel and you let it fizz up too much above the level of the gel, it will get too hard to work with. And what you'll find is that the DNA fragments don't move through it easily, and that you basically have wasted a gel dish, because when you go to take the forks out, they won't even come out properly if you overheat the gel. So you want to follow those directions specifically. And again, you're going to want to wait till it's cooled before you add in your buffer and your DNA. Why? Because it would denature, right? So now the cooled gel is transferred again over to that electrophoresis chamber. The gel is submerged in a buffer to add ions for the electric current to pass through. And again, that buffer is going to be something called TBE, trisborate, and then EDTA. Okay, and then the samples are added into those wells. And again, you're doing it very gently. You don't want to puncture anything. You want to go slow. And then voltage is added into the electrophoresis chamber, again, containing that gel and buffer from that electrode, the cathode to the anode. And then the DNA is allowed to kind of move. So DNA and DNA fragments, again, are negatively charged. So the current is generated by the negative and positive electrodes. So again, that negatively charged molecule, like a DNA fragment, is going to move towards that positively charged anode away from the cathode. And you can kind of see that demonstration again here. So what is typically added to gel? So again, we have a DNA ladder. It has numerous fragments of specific lengths. The lengths are measured in base pairs. Okay, and it's used as a comparison tool for determining the approximate size of the experimental sample fragments. And for our lab, we are going to have 10 fragments in our DNA ladder that range between 100 to 1,000 base pairs in length. The negative control, and this is going to be all necessary components needed for PCR, but with water as the DNA sample. Why would that be a negative control? And again, if anything happens with the internet connection, speak up so that you can hear me and I can hear you. And I know that if my internet connection goes out, sometimes I can still hear you guys tell me when the internet goes out, even if you can't hear me, which is weird. So let's think about this for a moment. Why would we want all those necessary components, right? For that gel added in, right? But without the DNA. Again, what is one of the, in the last couple of labs we've done and we've talked about, what is one of the largest confounding variables that can happen whenever you're working with DNA? Contamination. Yep, contamination. So what we're looking for is contamination. Because if we see lines on that negative control part of this assay, then we are going to know that our samples are contaminated and we cannot interpret the results, right? Because we should not have any results at all in that segment. And if we do, that means that contamination has happened. And the positive control, it's going to be all necessary components needed for PCR, but with a known DNA sample. For our lab, it's going to be the DNA sample from a heterozygous individual. If you remember last week, we talked about that the per 3-5 and then per 3-4. Per 3-4 was the per 3-4 is daytime, per 3-5 is nighttime. You can be homozygous for both, okay, and have either or you can be heterozygous. Remember how when we ran the lab we did earlier in the semester and we were talking about it and it was like, okay, so it would make sense in most situations to have this be a positive control, but this serves as a great positive control because it allows for a basis of comparison to see the possible results of the experiment, right? At both ends, whether we are on the low end or the high end of what we're trying to observe. In this case, being shorter or longer base pair lengths, okay? So we would see 2 bands with this, and that's the biggest reason why. So we could compare and see what it would look like if someone was per 3 homozygous, per 3-4 homozygous, per 3-5 homozygous, or heterozygous for the traits. Okay, and the experimental sample will be the unknown sample. So when we're looking at this gel, we're not going to expect any banding for the negative control. The positive control, we should expect 2 bands per lane, and the experimental sample is going to be based on what we predict. And if you remember, I scored a 32, so that means that I would be seen as being a what? Nighttime person, right? Or a night owl. So the results that you're going to be using to interpret your lab reports is going to be based on that. And I'll kind of show you what that would look like on a gel plate. And I do have actual results from gels that students have ran in class if you're curious at looking at those, but yeah. So again, the setup for this experiment, so that basically we would want you to have some experience pipetting in a gel well, which is why that we would have wanted you to pipette this so much. But the other thing is, right, like if we have 2 negative control samples, 2 positive control samples, again, if there's contamination, it gives us a better opportunity to be able to see that, right? And sometimes with banding, we can get something called smearing on gel plates. Have any of you guys ever heard of that before? Smearing on gel plates? So basically, it's whenever a band is so dark, when you look at the gel plate, you can kind of see like a ray of light that's much thicker than where it's supposed to be. And it almost looks like if you took your finger and you dipped it in some ketchup and you smeared it across the countertop. There's a lighter section and a darker section, and sometimes it can make it harder to read. And that's the reason why you put so many samples in, right? And you've got your DNA ladder on the both ends so you can kind of see that, right, as a basis of comparison. So visualizing the fragments, okay? Gels are stained with DNA binding dyes that when placed under UV light, they will glow. And this allows us to look at the DNA present at differing locations along the length of a gel. A typical DNA stain is something called ethidium bromide. You remember how at the start of the semester I was telling you guys, you've got to get used to being able to do this, even if we don't necessarily use the toxic chemicals. The toxic chemical that I was most concerned about you getting used to being able to be in contact with without getting contaminated is ethidium bromide. Even though we were going to be working with something called sea green, having really good lab etiquette whenever you're running gels is really important, because a lot of times you will come in contact with something called ethidium bromide, and ethidium bromide is noxiously cancerous and a big irritant. It's not something that you want on your skin. And it's to the point where, like, Dr. Estep, when he runs his labs and he does his DNA analyses, the entire countertop is covered in ethidium bromide. And, like, they don't want you to touch anything on the counter until it's been decontaminated every semester at the end of the semester. It's that big of a deal. So when you are working with things like this in the future as you move forward, even though I know you may not have the experience of working with sea green, if you ever get in a lab where you are working with ethidium bromide, please be careful not to get it on you, okay? Because it's very toxic. And you can kind of see that visualization here, ethidium bromide. That's its abbreviation. So now that we've talked a little bit about the gels, I want everyone to kind of unmute themselves but remain quiet if possible, and try and take a shot at looking at these samples and telling me what these base pair lengths are. So can anyone tell me what the length of this segment is here? Based on comparing sample A to the marker? Ideas?
You just went out.
Okay. Oh, it says I'm still screen sharing. Okay. All right. But can you guys tell me what this base pair length would be?
Is it 1,000?
Yes. And what about this one? 900, right? And then this one. This is a little bit harder, right?
700?
It would be about 700, yeah. And then we have this one. What would that be?
450?
That'd be about 450. And then here, it's a little bit less than what?
Less than 200.
Yeah. Can anyone tell me the length here?
Isn't that one still 1,000?
Yeah, and so this one would be a little bit greater than 1,000 actually here. But this would be about, I want to say like 825, right? You could look at this, and this would be about 275, and this would be about what? 75, right? And anyone want to try this band here?
Maybe like a little lower than 600.
It would actually be a little bit greater than 600, because you got to remember, the closer we are to the top, the greater that base pair length, right? Because the larger base pairs don't move as far. So it would be a little over 600. What about this one here? A little over 5. And then this guy. Anyone want to give me an idea about what he might likely be?
Look at the light.
Like maybe
Look at the girl.
275, 250?
Look at the light.
275, 250. Pretty good. Okay, so determining our alleles. So we can take advantage of the different lengths of the alleles. Specifically, of our amplified PER3 gene, including 4 or 5 tandem repeats when we're looking at this process. So we're running our copies of these amplified genes on a gel that separates them by size, and we're using that ladder to estimate the size of the fragments and then determine if the DNA included is only the allele with the 4 tandem repeats. So would you see a really dark band in one spot, that would show that someone has daytime tendencies, or morning time tendencies. And then with the 5 tandem repeats, we'd see a really dark band at the nighttime tendencies, right? And then a mixture at both 2 lighter bands at each of those subsequent locations, okay? So I know this is a big screen, but we're going to talk about it again. So again, the 2 copies of that PER3 gene with those 4 tandem repeats are homozygous for PER3. Okay, they're 2 identical alleles, and variation is thought to be associated with being a night owl, and it's represented at 250 base pairs. Okay, and then when we have 2 copies with the 5 tandem repeats are homozygous for PER3-5, we have those 2 identical alleles of the PER3 gene, and variation is thought to be associated with being a morning lark. And again, that would be present at 300 base pairs. And then when we're heterozygous, we have 1 of each of these bands in a lighter color, again, represented at 250 and 300 base pairs. So based on this, I kind of want you guys to come up with the answers to this. I know this is kind of hard, right? What is the experimental question? So if we remember correctly, right, I scored 32, so we would think that I was a nighttime person, right? So if we are looking at the base pairs, right, and we're looking at, and we would think homozygous nighttime, right, or homozygous evening time, you could say that your hypothesis for the experiment, and you would really want to clean this up, is that your instructor would be homozygous for the PER3-4 gene at 250 base pairs, okay? So, yeah. Or, yeah, that would kind of be your thoughts here, where you kind of would want to head. So your experimental question, right? Like, what is your instructor's genome in comparison with the provided information, right, based on the survey, and your hypothesis. Okay, so you'd have to have an alternative and a null hypothesis. Your null hypothesis would be that what? I could be homozygous for traits for morning time or heterozygous for traits for morning and nighttime, and you'd want to talk about the alleles. And the alternative hypothesis would be what? That I would be homozygous for the nighttime variation of the alleles, right? And independent and dependent variables. Again, so independent, right, and dependent. So, yep. Independent you could look at it as like your study subject, right? Like your individual instructor. And dependent variables would be what? Your individual base pair lengths. Okay? So the genome associated with that. But you definitely want to clean those up when you're writing those into your notebook. And your phenomenon of interest. In other words, what do you expect to see happen in your positive control but not in the negative? Right? Yeah. What do you expect to see happening in your positive control but not the negative? Well, we shouldn't see anything in the negative control, right? Because we're only including the ingredients for PCR and water. And in your positive control, we'd expect to see what? Heterozygous traits as a basis of comparison for someone who is either homozygous for nighttime, daytime, or heterozygous for both traits in the tandem repeats of the PER3 gene, right? So I want you guys to kind of talk to one another as a class for a second and just try and come up with a deductive logic statement. I know there's a lot of voices, but if anyone has any good ideas for this deductive logic statement, throw them out, even if you're not sure, okay? Right, we're talking about negative and positive controls, and you could start with if there is good working order for the experiment, then, so I'll leave the prediction part to you. If the experiment is in good working order, then I don't want to call out names. I do have you guys' names memorized. Let's see. Sam? Do you have an idea?
Well, quite frankly, I'm trying to. What was the experimental question? Because I didn't quite catch it.
Well, the experimental question is just trying to determine basically like your instructor's genome based off of the provided information with the DNA analysis. So the experimental question would basically be that, just kind of trying to find that.
Okay.
So
If there is good working order, then we shouldn't be seeing any bands in the water sample, while we should be seeing bands in the non-water sample, DNA sample. If I'm
yeah,
understanding this correctly.
you are understanding it correctly, which that was great, by the way. So for the negative control, right? So if there is good working order for the experiment, then we would see no bands for the negative control group. And for the positive control, we would see bands at 250 and 300 base pairs, representing the per3/4 gene and the per3/5 tandem repeat, or alleles of this gene, right? And so you could kind of write that down that way. So now, what would be a good control group for this experiment? Certainly not the negative control, right? The negative control has no banding. There's no banding with the negative control at all. So we can't compare anything to the experimental group with that. So that would leave the positive control. But why, apart from the negative control not being a good control group, would the positive control be a good control group? Ideas? Armstead, you got an idea?
So like, would a good control group be, say, like, is our not water sample the control group, or is that the negative control?
That's the negative control, right? Because it's soliciting no or minimal response. Okay? Because we're getting no banding. We should get no banding unless there's contamination within the negative control.
Okay. Huh. Yeah, I'm not sure what a good control group would be, but maybe. Sorry, I'm in Clemens right now.
You're
Maybe.
let me help you out a little bit.
Okay.
Yeah, let me help you out a little bit. So for this one, the positive control would serve as a good control group, right? Because we're heterozygous for that banding. So we're getting that per3/4 band and that per3/5 band. And that serves as a good basis of comparison when we're looking at someone who is homozygous for per3/4 or per3/5, or heterozygous for both genes at 250 and 300 base pairs. Because we can see those bandings apparent in that heterozygous individual. So that
Okay.
that would kind of help. And again, that'll answer that second question. So which group represents the experimental group? It should be your instructor's DNA, right? And that's because that's the result that we're trying to test to see, right? That's the DNA sample we're trying to test to find the result of. So, and for the null hypothesis of the experiment and alternative hypothesis. And so again, this should be a succinct comparison between the control group and experimental group in consideration of the null and alternative hypothesis. So, and you could talk about banding length here if you so chose. So for the null hypothesis, again, we're anticipating that I am a nighttime person, right? So the null hypothesis would state that I was not a nighttime person. So, and the alternative hypothesis would state that I was. And that's kind of the way you would want to word this, but in comparison of our control group, right? With that heterozygous banding. So, one thing that you could say is kind of roughly the prediction for the null hypothesis you could state that there is no apparent banding present at the 250 base pair sequence in the instructor sample as compared to the control group, which has banding at 250 and 300 base pairs. And then for the alternative hypothesis, you could state that, or the null hypothesis would state that there is banding present, right, at 250 base pairs, similar to that which is seen in the control group, which has 250, and then banding at 250 and 300 base pairs. And then for the alternative hypothesis, you could kind of state, banding was not present at 250 base pairs in the experimental group, but in the control group, banding was present at 250 and 300 base pairs. And again, you'd want to clean that up again. And again, you could talk about how that applies to me being a morning lark or a nighttime, or a morning person or a nighttime person again. So, and again, if the null alternative hypothesis, then prediction, sorry. That was more along the prediction that I gave you, sorry. So the statement for the null hypothesis would be more along the lines of there is no huge difference in between the banding that is seen for the control group as compared to the experimental group, in that this person is not a nighttime person. And then for the alternative hypothesis, it would be that there is a big difference in the banding, and that, this person is a nighttime person. And then you could talk about what I had just talked about. That would kind of help you a little bit. So for the banding, this is an example. This isn't really what I want you to use, because this person is heterozygous for this trait. So you can see the DNA ladder here. You can see those positive control samples here, and you can see the banding present for this person, right? You have your positive control run in duplicates of 2 lanes, and your experimental sample run in duplicates of 2 lanes. And again, your base pairs are seen here. So now I kind of want to take a moment while I have you guys' attention and kind of just redirect you to the video so that you can watch that if you haven't. So this might actually help you process things that I've talked to you about a little bit more. And again, I may not have stated everything in the cleanest way. So whenever you are going through and working on your DLNs, make sure you're being really clean with the things that you state in your DLN. A lot more polished up. But let's go down here and let's kind of check this out. So, okay, where's the video? All right. I'm going to kind of pause this, and I just want you guys to I'm going to stop the share. And then I'm going to re-screen share. And when I do this time, I'm going to enable the audio system so you can kind of see.
Hello and welcome to Exercise 7, Uncovering the Results of DNA Analysis in Lab Protocol video. Today, we're going to look at the results from our PCR, which we conducted last week. You may remember creating these PCR products and then freezing them last week. Today, we're going to take a look at the differences in our PER3 genes from our instructors by visualizing those PCR products utilizing gel electrophoresis. To get started, we need to prepare our gel. Our gel is created in this casting tray, which, in order to give it form while it's liquid, we set into this mold. Once in the mold, we need to add a comb, so-called comb, to allow for the gel to form around those comb teeth and create what we call wells, which are spaces where we will insert our PCR products before running our gels in the electrophoresis machine. To create the gel, we have to heat it up in the microwave. So come with me to the back of the classroom where we're going to dissolve our agarose and add it to our trays. To dissolve our agarose gel and prepare it for casting, we need to get our solid agarose tablets and add it to this Earl flask. Then we'll add 20 milliliters of deionized water, which I've pre-measured, into our flask. Then I'll swirl it around to fully dissolve that tablet. Now I'm going to need to heat it for 1 minute in the microwave. Now we're going to remove that from the microwave very carefully using some hot hands. And immediately pour the gel into our casting tray. We can give it a few taps to see if we can help release any bubbles that may have formed. And then immediately start a 10-minute timer. And when that timer's up, the gel should have fully cooled. All right. For the next step, you'll need to remove the comb so that the wells of the gel are open and ready for our samples to go in. To do this, you need a teammate to stabilize the tray so that the tray doesn't get removed as the comb gets removed. So I'm going to have our teammate stabilize this tray just like this, and then we're going to pull directly up on the comb, just like this, leaving those wells open. Next, to pull out the tray, your teammate's going to stabilize the casting form, and you're going to reach in and pull straight up like so. Now our gel is ready to go into our machine. Now that the gel has been removed from the casting tray, we're ready to run gel electrophoresis. The electrophoresis chamber is going to be set up in the following way. The base, which should be unplugged, is ready to go. Then we add the buffer chamber into the base, making sure the notches line up in the correct way. Next, we're going to put the gel tray into the buffer chamber, just like this, making sure everything is lined up correctly and you're not forcing anything. Now we're going to obtain our conical tube, which has 30 milliliters of TBE buffer solution. And we're going to gently pour the buffer solution over the surface of the gel that's in the buffer chamber. This buffer should just cover up the gel and the wells, and we should make sure that there's no air bubbles in those wells. So gently pouring over top, like so. If we do notice any bubbles, we can just gently tap on the buffer chamber to release those bubbles and help them come to the surface. Next, we're going to find our micropipette, make sure that it's set for 12 microliters. And we're going to begin by filling our wells following the instructions and the diagram found in the lab protocol. The diagram shows us where we want each of the different solutions to go and each of our different samples to go. And will indicate to you where you put your ladder, your negative control, positive control, and experimental sample. The first well is going to contain our DNA ladder. We get a fresh pipette tip, open up our ladder here, obtain 12 microliters, close it back up, Don't want to spill it. And dispense those 12 microliters into the well. As you do this, you want to stabilize your elbow on the table so it's nice and secure. Make sure no one's shaking the table. You can use your second hand for extra stability. And then you're going to place the pipette tip into the well. Now, you should have practiced this beforehand, but you want to make sure that you are not puncturing the well, puncturing the gel, and that you're not introducing any bubbles. You want to dispense very slowly. And don't release the plunger until you've removed the pipette tip from the solution. Then you can discard that tip and move on to your next solution. The next one will be your negative control. Get our negative control here. 12 microliters of this. Again, stabilize your elbow, stabilize your hand. Don't puncture the gel. Dispense, but don't release until out of the buffer. We'll do a second well with the negative control. Then we'll move on to our positive control, where we'll do 2 wells with the positive control. Finally, we'll do 2 wells of our experimental sample. We want to then put 1 more well of our DNA ladder. Now, with your wells filled, you're almost ready to run your gel. Before you place the cover on top of your machine, you'll want to make sure that you have added this clear view spray to prevent fogging. Give it a spray, rub, so there's no streaks. Then your cover is ready. We'll place the cover onto the machine very carefully, nice and slowly. We don't want to disturb the products that are in the sample. Then we can plug in our machine, turn it on, and begin our timer. We'll come back in 45 minutes. All right, Now that 45 minutes has passed, we're going to apply our viewing hood or viewing cover. You're going to unfold it like this. Now, when we view this, we have to use our smartphone. We cannot look directly at it. It will harm our eyes. So we'll cover this up like so. Turn off our machine now that 45 minutes is up. Turn on the light, and we're going to just zoom in here. And take a look. This is a great, we've got pretty great separation now after that 45 minutes has passed. We're going to take a look. We can see our ladders on either end. Our negative control, our blank, positive control, and our experimental sample. You'll discuss with your instructor what this gel is showing. All right, we're going to just take a quick look at our gel electrophoresis results. We're going to see examples of all of the three.
Now, I hope you guys can hear me now. All right. Okay. If you end up having to dip out, Sam, because of the internet, that's completely okay. Now here are kind of the gel electrophoresis results. So here you can see someone who is heterozygous. We've got that ladder, we've got those negative controls and these two wells here with no results because, again, we're looking at contamination. Positive controls, and you can see distinct banding at 250 base pairs and 300 base pairs here. So that's what a heterozygous person would look like. Homozygous for PER3-5, this would be someone who has the morning-like characteristics. And again, you've got that ladder here, this empty space where the banding is not present, right? And you've got that negative control where the banding is not present because it's water, and then the PCR recipients, right? Just checking too. Yeah, all right. And then we've got that positive control, and you can see distinct banding that lines up here at 300 base pairs. And then we have homozygous for PER3. So here we have that ladder. Again, we have that distinct negative, right? And here we only have one result for someone who is a homozygous for the PER3-4. If you kind of notice, even though these lines are a bit wavy, if you look here, it kind of lines up a bit more. There's a bit of smearing here, but it lines up more at 300 base pairs for the PER3-4 gene. So for your lab reports, the one that I want you to use is the one that is associated with nighttime behavior. Again, it's just double checking again, that way I don't tell you the wrong thing, but I'm pretty sure it's the PER3-4. Just to double check, let's double check it again. Yeah. So yeah, night owl, PER3-4. So when you're writing your lab reports, you kind of want to include this gel result as your instructor gel results when you're working through those. Do we have any questions at all from anyone? Any confusion so far? Yeah, go ahead and ask.
How do we get the results for the hypothesis or the null hypothesis, like to prove if it's valid or not?
So basically, the null hypothesis is going to kind of state that the results are going to look more like a heterozygous person, or it's going to look more like a homozygous for PER3-5. And that's kind of how you would want to phrase that. And if those results do not look like that, the null hypothesis would be found to be invalid. So, which they don't, because they look like nighttime. So, because you would be using those nighttime results again. Any other questions about this so far? Because I do have a couple of other things I do want to go over with you guys very briefly.
Yeah, are we like still using like your DNA for like the results, or are we like
Yes.
in the video?
You would want to use that nighttime, right? So those nighttime tandem repeats that are associated with that behavior whenever you're writing up these results. Yeah.
How are we like gonna get the results from yours though?
That you would just pretend that this was mine. So homozygous for
Oh,
periphery 4. Yeah, that's what you would do. You would
okay.
be like, okay, this is what the gel would look like. Yeah. Any other questions about any of this so far? Because there are a couple of other things I do want to talk to you guys about. So now in the back to the slides, so basically there is an exercise 6 technique certification, which is an analysis of DNA, which is interpreting gels with the CSI wildlife. So basically here you would be kind of looking at seized ivory and looking at the genetic analysis of that and mapping off different areas based on that. And you would kind of want to get this done at the same time this Sunday. And then you have your genetic disease module project. So there are some instructions for completing this genetic disease project that are available on As You Learn you can check out. You can kind of follow this along pretty carefully. And then the presentation slides for that are due Sunday, November 17th. And the genetic disease presentation peer review of these slides are due Friday, November 22nd. And the genetic disease presentation recording is due Tuesday, November 26th. I will say, you can pick out your genes and you can do your project in such a way as this, but you can also use FASTA and BLAST in order to get those peer review articles, especially if you're having a hard time looking at them for a certain genetic disease. You can follow the instructions such as this, or you could do it this way. I think doing it this way makes it easier. If I'm not mistaken, we're going to start with FASTA. You have to give me a second. Yeah. Format for nucleotide sequences. So here we go. And one more thing. Give me a second. One thing. Okay. So let's go here. This is one of the websites that you can use if you so choose to. Give me 1 second when I talk to you about this. Here, I'm going to break the screen share for just a second. If everyone can give me just one moment so that I can go in and see if I can find this recording that I'm looking for. I took of myself a while back. Because there is one where I talk about, and I did this last time I taught this class because it was just super easy for everyone to do it this way. Give me 1 minute. All right, here we go. So you can use FASTA, and you can also use a website called OMIM. It's omim.org, my bad. And let's just go to that real quick so I can kind of show you around. And this one's kind of cool. So this is a website that you're going to get to use a lot more later on. But if you're looking up, so can someone just name like a random genetic disease that they would probably be interested in looking at for their project?
Sickle
Because this
cell?
is all, huh?
Sickle
Okay.
cell?
Let's look at sickle cell anemia. Okay, and let's look it up on OMIM. So basically, this pulls up all of the cytogenic genes associated with sickle cell anemia and all the research of genes associated with sickle cell anemia. Here we can see this first entry, and this is ranked number 1, so that would be the one we want to click on. And here it gives you the location on the chromosome, the phenotype, right? The phenotype number, the inheritance, okay? It's autosomal recessive. The phenotype mapping key, which shows you where along that molecular basis it is known to exist, the gene and locus name, and the gene locus and mem number. So if we go down here, and we're looking at some of these diseases, it can give us clinical features that we can use to write our papers with, a general description of the disease that we could utilize in our PowerPoint. Do not copy things word by word. Diagnoses, clinical management of the disease, and all of the most modern research associated with that disease. The pathogenesis or the mode of inheritance and how it affects the body, the molecular genetics associated with it, the population level genetics associated with it. It'll even give you an animal model to use as reference because it's a disease that you would see in people. And then here, something even cooler with this is you can click on this location, right? And you can see all of the phenotypes of this gene that are associated with it. And you can kind of see how it even is associated with malaria resistance. Certain, genes associated with this are. And then you have your phenotype mapping key. I wouldn't mess with this part too much. I would kind of just focus on the other parts of OMIM. So let's see. Let's look at this from a linear model. And I don't want you to have to look at it that way, actually. So gene locus mem number. Yep. So, yep, here is where you would kind of want to write down all the phenotypes that can be associated with this gene. And you can look at each one of these with this gene location and changes in that inheritance and how they can affect not just the formation of sickle cell anemia, but things like malaria resistance that kind of go hand in hand with it. I think that this is a good starting place. And then here you can even find allelic variants or differential variants of the gene and some information about that. And again, the references here are always great whenever you're trying to find articles that give you information on treatments, right? Because we have an entire, area of that. So like you could look through and you could see if any of these are treatments, specifically epigenetic treatments. So that's a great way to find some different things. And then here, if you go into GWAS, you can get like the total genome association of that study that's associated with it. If you click down to DNA, it'll give you the NCBI reference sequence, which is kind of cool. And I'm going to show you guys how to kind of do something with this. So this is the gene, right? And you can kind of scroll down, down, down. Maybe not. Here we go. Hold up. Or you can kind of click here and you can right. Kind of hit show in the genome data viewer. And you can get actual images of this gene to put in your PowerPoint slides. and its location. You're just going to want to kind of check over back to OMIM and make sure that it's referencing that same exact gene where you're looking at it. And you can kind of include that in PowerPoint slides and give me some kind of information about it. It's even got the more highlighted versions up here that are a little bit easier to follow along with. But you can see how it says chromosome 11 and it's giving you some details about the gene. And yeah. So again, you know, that's a great way to find information. And if you want to go that route, you can even look up disorders and choose something that's a little bit less traditional. And you even got your human protein atlas, which is really cool here. So like here, it can show you where it's expressed at in the body, if that's something that you're interested in knowing. And uniprot and then NCBI protein. And then if you click DNA, right, I think we did that already. Genome, clinical resources. So let's see, just check this out. Yep. And then you can get even more resources and genetic and rare disease information resources, and then clinical trials of medications and different things that you might be able to find information about with all the references put in. So I think that this is a way that will kind of make it easier just to find information for you, just to kind of get used to playing around. So I mean, like, for example, what's another disease that you guys might be interested in looking at? Maybe even something that has more or less non-traditional modes of inheritance even. Right? Any ideas of anything you could look up? You can even look up diseases like and you can look at one specific gene that's associated with its inheritance, right? So here's a gene, and if you wanted to focus on this gene's manifestation of this disorder, you can talk about how it's a biopsychosocial model, and you can get information that way. Like with the gene mapping and then the molecular genetics, you can find references and the phenotype gene relationship. You can find all those phenotypes associated with it, you know, the susceptibility to it. Right. So, right. And this is the one we're looking at, right? And the different forms of it that can impact and where that gene was first isolated from and that phenotype mapping key. And again, that inheritance here is not as common, right? This would be something that would be more along the lines of something like epigenetics, right? And then you could find clinical trial information. I would stick to something that's probably autosomal recessive or autosomal dominant for this. But I mean, if you can find different kinds of things that you want to talk about and you could find enough support for it. And I will say, if you're going to choose a disease like something like schizophrenia, I would want you to probably choose multiple genes to talk about because they may not all have the same inheritance patterns, right? Like, and you might not be able to get as much information off of one versus another. So you could click over here and check this out. And here, autosomal dominant. And it even has the gene locus information. And you got to think, so they're adding to this database as soon as they get more scientific information to kind of assist you with finding things. Huntington's disease even is a cool one to check out on OMIM. It's just a way for you to like kind of play with it, right? And then you've got, right, all these different genes, right? Autosomal dominant. You can kind of find information on those, or breast cancer. Right. And you can find individual genes that are associated with the formulation of breast cancer. So, I mean, if that's, and again, you can also narrow these down to specific kinds and get information that way. What's a common one that's got multiple gene control? Here, let's go. Look, okay. Major affective disorder, right? And then you've got all these different genes associated with it. And again, the inheritance for that autosomal dominant, and you can find different informations about it and talk about it. And again, if you're going to talk about a disease like this that is under multiple gene control and has such heavy influence from the environment, I would choose multiple genes in the PowerPoint presentation. But I mean, besides that, it's a great way to find certain resources. It's just starting with a website like OMIM or BLAST or FASTA or NCBI. NCBI is a great resource too. Let's just check this out real quick. So let's go in and let's look at the genome data viewer, right? And we're like, it's pulling us back up. So we go to home here. Common or scientific name for organisms, right? Genome, location, gene, or phenotype. Let's see what we might want to look at on NCBI. You had said sickle cell anemia before, right? Right. So you can just kind of hit search. And this is one of those websites that takes a while to load because the more people there are on the browser, the harder it is to load. So just give it a minute. Yep. And so these are the genes that are associated with it. We can click here and get some information about this. And again, you can do the same thing in here. You can go in, you can see that specific chromosome length, and you can do the same thing where you can get the gene name and or the gene location name, and you can kind of type it in here and get some of the information that way, or you can blast it. And right. So you got that view again. Pretty sure there's a way to do this in a different manner. Give me a second. Okay. Okay. All right. I guess not. I lied. Yep. Just give it a second. But yeah, I mean, that's a great place to start getting information about it. Right. You can kind of scroll down here where it says blast a genome. And let's do that again. It's got that accession number on there too. That might be associated with the disease, right? And it might give you more information about it that way. So it's just a great way to find information if you need to know something about a specific disorder or a specific disease. So, yep, here we go. But yeah, so let's say we looked up sickle cell anemia. It gave us that reference sequence. We could kind of click here again, kind of check that out. And then go in and it gives us all this information, and then we can click the GenBank accession number that we're most interested in, and it gives us literally everything. So if you scroll down here, you can even let's see. Yeah, this is for proteins, but right there. But yeah, it can give you like the primary article where it was found and what it's associated with. So you should be able to get the full genome. But yeah, let's do this one more time. Here we go. Shows you where it's at on the chromosome and all the information associated with it. And again, that's just something really cool, right? Like you can get a lot of the information that you need. If I can find the video where I kind of go into more detail about this, I'll post that on YouTube and you guys can follow along with that and kind of use it as help. And you don't necessarily have to use these tools. And I know it can be like, oh gosh, she wants us to do something terribly hard. It's really not that difficult. I mean, you're kind of just like, plugging information in, right? And like finding all what you can about it. And then here this says about article, more so, but then you got your origin. You can use the accession number that it gives you to kind of do that. And if you click here, if you're looking for something in particular, you can look at FASTA and it'll give you like the literal entire genome sequence if like you want that for something. And again, you just got to make sure that you're using that same GenBank number. You can even look up the GenBank number on OMIM and then type it in here and it'll pull up every single article that's associated with it. And then you can hit FASTA and get your entire genome. I mean, like you could really work through this and get like the entire genome, an image of like where it's located at on the chromosome if you wanted to, all of the genetic information associated with it. It's just a really great way to like get access to like information. So yeah, which is just kind of cool. So, but like if you wanted to do that, say we went to like, here, let's just go back to omim.org. And let's start with like, I don't know, let's look at Huntington's disease. I think that was the example I used last year. You can kind of click in here, right? And then it gives our gene location or phenotype number. And then you can find mapping, molecular genetics, references. So, yeah. Here, let's click down on the next one down actually, because it might have more of what I'm kind of trying to show you can do. Yep, here we go. You can kind of click here and you can get that protein and gene info, and you can get a human protein atlas and get information about like what the proteins look like and what it does and like all the coding genes associated with it. Like really, if you just play through here, you can get so much information. And, it's just a great resource to have whenever you're looking up genetic diseases. And again, I'll try and like find that video where I go into more detail about like ways you guys can use these in succinct with one another or like, in total with one another. But I mean, in general, if you're just looking for like a place to start, OMIM, NCBI, and like, this is just a great place to start to find information. And let's see. Yeah, Gene. Boom. Let's say we wanted to find HDL. Okay, Control C. See, we go in here and we kind of just boop and then hit it and then hit search. This is going to give us our model species. And then it's like all these articles and all these resources associated with it and all this information about it. You can just click straight through. And so, and find more information about the phenotypes. And again, that resources is straight from OMIM and then protein products, right? And then you've got that information there and interactants, other genes it interacts with. So you could go here and click phenotypes. And again, you can click on this should be able to. Yeah, like, I mean, if you find like one particular disease and like have the gene in general that you're interested in, it can give you so much information. And then if you go to ref sequence, it'll even give you like, hold up. Yep. That's the related site. Sorry about that. But yeah, you can get the information from it. So it's just kind of cool. So I just wanted to kind of show you guys this. Because, I mean, if you're looking here, you have all these articles, right? Localization of the gene for a notable autosomal recessive neurodegenerative Huntington-like disorder. Vascular proteoglycan binds differently to HDL2 and HDL3. And that's a great way for you to find like epigenetic or like genetic therapies, right? Cross-linking modifications of HDL APOP proteins by oxidizing phospholipids. And their functional implications. So, yeah, if you look through those, I'm sure you would find something. So, but yeah, I mean, it's just a great way to like find the information about it. So, and again, you just go in and find it and click back through. Because, I mean, all these websites are kind of just like connected with one another, which is a great thing about it. So, yeah. It'll even give you like information about things that they interact with, which is super cool. So, I mean, yeah, it's pretty dope. Again, I'll see if I can't post that video and see if it's just a little bit more help to you guys if you guys are looking to do something kind of like interesting. But this is especially helpful if, like, say, for example, like you go on OMIM and you're looking at something like, I don't know, schizophrenia, and you're like, well, I want to do something different, you know, because everyone else is going to do something that's like autosomal dominant, autosomal recessive. And you want to like learn about the specific gene, you could just pop it straight in here after you've changed it to gene and kind of just like get information based on it that way. And you got that gene name, right? And then you've got, not for this one. You got to be a little bit careful about it. So not every single gene you'll have information, a lot of information about. But yeah. But yeah. I think this is what it's asking me for because I think this is the same thing. Yeah, it's just a different name for the same thing. It's got two names. But yeah, it's protein coding, talks about all the organisms that have it, gives descriptions about it, and like how it's related to psychiatric disorders and like so much information. And this is like really helpful if you, again, are like wanting to do something a little bit different. And again, it even shows you like which part of the bodies it's expressed in, which is like kind of cool. And again, related articles to find information about it. And then gene references, right? Articles that talk about how that has been found. And if you're looking, you know, for information, kind of like more along the lines of gene therapy, anything with CRISPR probably will have that. Right. And some of these genes, even like all alternations with them have been associated with like different, you know, phenotypic diseases and how they manifest themselves. So then here it kind of shows you every single gene that it interacts with. It's really cool. So, and again, kind of go here, transcripts and products. Again, you've got that image of the gene. You can kind of get for yourself, which is really cool. So, yeah. See, NCBI reference sequences. Yep. And then you just kind of come down here and it'll give you those mRNA and protein sequence, right? And you can see information about those there. Just make sure that you're clicking on the one that's actually associated with like the accession number, you know, for the specific form of the disease that you're looking at. But it is really, really cool. So, and again, you can do a lot of things with the accession number of a gene. So, which is pretty cool. But Yeah. And it even has links to like PubMed, right? Down here. So that's kind of cool. But yeah. It's just an idea, something to play around with. I can post the other videos so that you kind of got a little bit more information as you're working through things and feel a little bit more comfortable with what you're doing. But yeah, let's see if I can find that video of myself kind of talking through it. But any other questions? Like I said, I just feel like it's a really fun way for people to be able to get information. Like I feel like it's a really great way. Every single student that I had who last semester used like GenBank and Blast and NCBI and like OMIM to get like the information for their final presentation. When they finished, they all had like 100s on everything. So I'll see if I can't get that video for you guys because it's a great way to get access to all the information that the project outline is asking you to get, but especially with the clinical trials and gene therapies and stuff like that. So any questions?
For the DLN, it says we need a picture. What should we do for that?
You probably are going to want to, I'll have to actually upload it to AssyLearn so that you guys can work through that, but it would be the one, they'd be that per 3-4. Yeah.
Okay.
So yeah, and you just want to make a figure out of it. And remember, you're going to want to label the ladder, the negative controls, positive controls, and the experimental samples at the top. And put that title down at the bottom. Other questions?
I have a question.
Mhm.
Is the peer review random?
For the assignments? It probably will be for this because, I mean, you got to think we're online right now. So it probably will be. You're talking about for like groups or something?
I just saw that it was like that for the project.
Do they have you guys working on it in groups or individually right now? Because in the past, they've had students do it individually. That's why I'm wondering. If there's a peer review for the project, you could contact your old group mates and work on it with them. If it says that you can work on it in groups, then you could do it that way, and we could peer review you that way. You just have to send me all your group mates and their information with their emails and stuff. But I'm pretty sure you guys are working on each one of these individually, and I think the peer review might be associated with like an end of semester, like how did your group do? Which, you know, we can just have that random if you guys want to. That's fine because you guys have been working with kind of random group mates throughout the semester. So what do you think? Any other questions? Anything at all? So when you submit the module project, you would kind of be recording yourself talking over the PowerPoint slides, if I'm not mistaken, kind of like through Zoom. So if you guys don't know how, you'll probably want to like go down here where it says more, and then you'll kind of hit record and then take it to the cloud. But any other questions? If you guys don't have any questions, you can go ahead and head out. Oh yeah, I'll definitely be doing that. I'm pretty sure I have a outline that I made at some point for all the students to kind of follow along with. So yeah. I'll just have to look through my docs. But anyway, you guys have a good day.